Hey guys, welcome back to my YouTube channel, and today is the first video we'll be doing, and in this video I'll be covering Unit 1, Cell Structure and Organization. Before actually moving on to Unit 1, I'll be showing you guys the syllabus content so that you guys would have a quick idea about what will be covered. If you think this is necessary, you may pause the video and take a quick screenshot and self-check at the end of the video to see if all parts have been covered. Moving on to the actual lesson, which is cell structure and organization, the first thing we have to know is what a cell is. In simple terms, a cell is the smallest structural and functional unit of life. As you know, there are many types of cells, and each of these cells have its own specific functions. For instance, red blood cells transport oxygen, nerve cells or neurons transmit nerve impulses, etc. All living things actually have cells and is categorized into two: animal cells and plant cells. And again, according to the amount of cells in an organism, this is categorized again into two: unicellular organisms, as the name suggests, one-celled organisms, which include amoeba, paramecium, yeast, basically all the organisms that aren't visible to the naked eye, and multicellular organisms, as the name suggests, many-celled organisms, which includes a large variety of all the visible organisms to us. As you all know, cells are extremely tiny, so it needs to be observed under a microscope, which could be a light microscope or an electron microscope. And once observed, you would see the cells as shown below, where on the left-hand side, you can see the animal cell, and on the right-hand side, you can see the plant cell. You have to have a great understanding about the parts of each of these cells, because in the practical paper, you might sometimes get a question to draw and label the cell associated with the magnification question. But overall, even in the MCQ paper, you may get questions to underline about the functions of each part, or which part is present in a plant cell and which is not in an animal cell. So have a quick understanding: animal cell, cell membrane, mitochondria, cytoplasm, which fills the entire cell, the nucleus and the nuclear membrane, and small vacuoles. In most cases, animal cells do not have vacuoles, but in some cases, there will be small vacuoles present. When moving on to the plant cell, it always has a cell wall, then the cell membrane, then the cytoplasm, which again fills the entire cell, the nucleus and the nuclear membrane. What is to be noted is that they have large vacuoles compared to animal cells that take at least half of the space of the cell, and an additional thing, which is chloroplast. Mitochondria again is there, as it's known as the powerhouse of the cell, and you may even find starch grains in the chloroplasts. Moving on to the parts of the cell, the features and its functions. First thing, cell wall, only present in plant cells, made of cellulose, and is a fully permeable membrane. For those who do not know what fully permeable is, fully permeable means it allows any and all substances to go through it. Moving on to the functions of the cell wall, it is the main thing that maintains the shape of the cell and maintains turgidity, that prevents the cell from bursting in when taking in water. Coming on to cell membrane, very thin layer of fat and protein, and is also called as semi-permeable membrane, selectively permeable membrane, or partially permeable membrane. These set of names come to this because it only allows certain set of substances to pass through it and not all. So hence the selectively permeable membrane. The functions of this is it encloses the cell and only some elements can enter, as I said earlier. Coming on to cytoplasm, which is a jelly-like structure containing 70% water. Metabolic reactions take place here and organelles are submerged in it. Functions include it holds cell organelles carry out different metabolic reactions. Vacuoles, found mainly in plant cells, and it contains a substance called cell sap, and the vacuole is made by a membrane called the tonoblast. Animal cells have small vacuoles, which are also known as vesicles. So the functions of this is to maintain the water balance and to keep the cell firm. Moving on to chloroplasts. Again, it is only present in plant cells, has a green pigment called chlorophyll. Photosynthesis takes place in the chloroplasts, and therefore you may find starch grains as well. So the main function of it is photosynthesis. Moving on to nucleus, which is the main organelle in a cell and is surrounded by the nuclear envelope or nuclear membrane. The nucleus contains the chromatin body, which contains the DNA, also known as deoxyribonucleic acid, which during cell division it divides and forms chromosomes. The nucleus also controls the quality of enzymes produced, regulates chemical changes, and determines the role of a cell. Moving on to functions, nucleus allows the transference of inherited characteristics from generation to generation, and the nucleus controls all the cell activities. Coming on to mitochondria, which is the oval rod-shaped structure, also known as the powerhouse of the cell, invisible to a light microscope, and more mitochondria is always present in places which need more energy. The only organism that doesn't have this is prokaryote category. Main functions of mitochondria is cellular respiration or aerobic respiration takes place in this organelle. The diagram here is given to have a brief understanding about the parts so that just in case a question comes, you have an overall idea that these are the parts in a mitochondria. Ribosomes, small organelle that is seen close to the endoplasmic reticulum or scattered freely, made of RNA or proteins. So main function of ribosome is to produce proteins or protein synthesis. For those who do not know what turgidity is, which was mentioned with the cell wall. So turgidity is the internal pressure in a plant cell when water is absorbed and the cell wall acting like a balloon. This keeps the plant stem firm and upright and the leaves flat. Moving on to differences between animal and plant cell. Plant cell has a cell wall made of cellulose outside the cell membrane. Animal cell, of course, no cell wall. Both animal and plant cells have cell membrane, nucleus, and cytoplasms. Only plant cells has chloroplasts that contain chlorophyll for starch production. Animal cells does not have chloroplasts. Plant cells have large vacuoles containing cell sac. Animal cells has small or no vacuoles. Plant cells have starch grains, but in animal cells there are no starch grains but may contain glycogen granules. Plant cell always more often regular in shape, animal cell irregular in shape due to the lack of the cell wall. Plant cell is around 40 to 100 micrometers in diameter and animal cell is around 10 to 20 micrometers in diameter. Moving on to specialization of cells, as I said before, most cells become specialized after cell division so that they do only one particular job. They develop a distinct shape and special kinds of chemical reactions take place in their cytoplasm. This specialization of cells is also called as the division of labor. We'll be talking about three main examples under specialization of cells, which should be root hair cells, xylem vessels, and red blood cells. Root hair cells, the outer part of the root hair cell, has a long tubular extension to increase the surface area for water and mineral absorption. Xylem vessels, made up of four main cells, xylem vessel cells, tracheates, parenchyma cells, and fibers. Xylem vessels are made up of a series of long xylem cells joined end to end. Once the growth has stopped, the end walls of the cell is digested, forming hollow tubes of joint cells. Then a substance called lignin, which is a complex organic polymer, gets deposited in the cell walls, making them strong and impermeable, which of course eventually kills the xylem cell. Lignin is formed in three distinctive patterns: spiral, ladder, or reticulate. Functions of the xylem vessel is to transport water and minerals from the roots to each and every leaf in the plant and to provide mechanical support to the plant. Coming on to red blood cells, very small, about 7 micrometers in diameter and 2 micrometers thick. It's a disc-shaped biconcave cell that is red due to the presence of the red pigment hemoglobin. Hemoglobin readily combines with oxygen, forming oxyhemoglobin, which is transported throughout the body. The nucleus is not seen in red blood cells in order to provide more space for oxygen transportation. And every cubic millimeter has about 4 to 6 million red blood cells, which are produced by the bone marrow and has a lifespan of about 120 days, 3 to 4 months, and is destroyed by specialized cells in the spleen and liver. Here are some additional examples of specialized cells. The ciliated cells are found in the lining of trachea and bronchi in the respiratory system, and its function is to move the mucus upwards and to act as a sort of filter in the air. Root hair cells near the end of plant roots, and its function is to absorb water and mineral salts. Xylem vessels found in stems, roots, and leaves of the plant to transport water and mineral salts as well as to act as support to the plant. Palisadian mesophyll cells found beneath the epidermis of the leaf, and its main function is photosynthesis. Nerve cells are found throughout the body of all animals, and its function is to transmit information in the form of electrical impulses. Red blood cells found in mammals for the transportation of oxygen. Sperm and egg cells found in testes and ovaries, respectively. These two fuse together to form a zygote. Lastly, coming down to cells and organisms, a large organism obviously contains millions of cells which specialize in carrying out activities which are characteristic of living things. A group of specialized cells that perform the same activity together forms a tissue. For instance, the stomach lining contains cells that produce digestive enzymes. The stomach muscle is also a tissue that helps in moving the stomach wall in and out, churning the food and mixing it with enzymes. Even in plants inside a leaf, for instance, when we consider palisade tissue, main function is for photosynthesis. Then organs, which are a group of tissues that work together to achieve a common task. For instance, heart, stomach, kidney, etc., all have multiple tissues working together to complete its final role. Onion bulb, leaf, stem are also organs found in plants. Coming on to organ systems, which are groups of organs working together to perform a specific task. For instance, the mouth, intestines, stomachs all together make up the digestive system. The heart and the arteries, veins are part of the circulatory system, etc. Organisms, a group of organ systems working together carrying out the characteristic activities of living things forms an organism. Overall, you have to have an idea that a single cell on its own is always specialized. So a group of such specialized cells is known as a tissue. Then a group of such specialized tissues is together known as an organ. And a group of organs that work together forms an organ system. And a group of organ systems that work together forms an organism. Coming on to the practical side of this unit, which includes the microscope and observing the animal and plant cell. Microscope, as you know, is an instrument used to observe extremely tiny things that cannot be seen by the naked eye. Generally, all specimens, animal, regardless of animal cells or plant cells, are usually stained with a dye in order to see the things and identify parts of the cell clearly. Methylene blue is used to stain animal cells, and iodine is generally used to stain plant cells for observation. As I told before, there are two types of microscopes, light microscope and electron microscope. The light microscope, as the name suggests, uses visible light for observations, whilst the electron microscope, as the name suggests, again uses a beam of electrons and can magnify an object up to 500,000 times. It's always essential for you to have a brief understanding about the parts of the microscope as it may be questioned in certain MCQs. So let's observe this diagram. The top part is the eyepiece lens. Then this entire part is known as the body tube or ocular tube. This is the revolving nose piece, or generally called nose piece. Then these would be the objective lenses, together known as objective lenses, but then according to the length of it, it is low power and high power. There's generally three objective lenses, although only two is shown here. This will be the stage, and these are the stage clips that holds the specimen slide. And below the stage is the diaphragm that controls the amount of light that enters the stage. And below that is the mirror, which reflects a beam of light, either from the sun or from an artificial lamp into the specimen. This is the base. The inclination joint is not an essential part that should be known. This is the arm connecting the ocular tube to the base. The two important things, coarse adjustment knob and the fine adjustment knob. Coarse adjustment knob moves the ocular tube up and down accordingly till the specimen slide is clearly visible. Fine adjustment knob is used to bring the specimen into sharp focus. So the two practical works that are included in this syllabus is the preparation of, or the observation rather, for plant cell, which is the onion cell. They have to peel an onion layer and observe it under the microscope. The observation would be as shown below. In conclusion, what is seen is the cell wall, cell membrane, vacuole, nucleus, and even starch grains can be seen in the onion cell when observed under the light microscope with a magnification of up to 400 times. The second practical activity is the observing an animal cell, where you scrape a cube of fresh liver by a spatula to gather a few cells and observe them under a light microscope. You would observe something like this, where the cell membrane and the nucleus is clearly visible with the sight. So in the conclusion, a cell membrane, cytoplasm, and the nucleus of animal liver can be observed under a light microscope with a magnification of 400 times. This is the most essential part, which is calculating magnification, always a frequent question in the practical paper. So these two equations, or rather the main equation, which is given here in bold, is the thing that you must remember, because in case of a magnification question, this equation will always carry at least 1 or 2 marks. Magnification equals to observed size of image over actual size of specimen. So we have come to the end of Unit 1. If you all think that this note which was used in this video was useful and would like to have a PDF copy of it, please visit the links mentioned in the description below. Thank you all for watching this video, and if you guys found out this video to be helpful, please do not forget to like, comment, and subscribe to my YouTube channel. It would be a great help. Thank you and have a pleasant day.